Okay, so I wanted to say this has been such a great conference. Thank you, organizers. It's really been my favorite online pandemic conference so far. Although maybe the bar is not that high, but really it's such a great selection of talks and really good conversations. So this is going to be a little story about data to models and back. And it's the first time I'm presenting this work, so I'm really interested to hear what everybody has to say about it. So I'm going to talk about shifts in mutational biases and how we found that they enhanced access to beneficial mutations in an E. coli study. So the mutation spectrum, you all know that the gain and loss of DNA repair enzymes, those gains and losses can change mutation rates. They're the famous, mutator phenotypes in experimental evolution. But these repair enzymes also make pretty dramatic changes to mutational biases. For example, they change the ratio of transitions to transversions, or they can dramatically change the ratio of GC to AT mutations or AT to GC. Here's a little sketch for the biochemically impaired, such as me. And bias can change the nucleotide or the codon or amino acid composition of genomes and proteomes, and that's been well studied. But what has received kind of less attention is, does the mutation spectrum affect evolutionary trajectories, and how so? How would it affect adaptation? It makes sense that the mutational spectrum affects the beneficial or the supply of the raw material for adaptation. And so we expect we might see large effects, but it hasn't actually been looked at very much. So this is work that I really can't take much credit for. This is a beautiful set of experiments that I'll describe to you by my collaborators, Deepa Gashi at the National Institute for Biological Sciences in Bangalore, and her former PhD student, Murdula Sane, who's recently moved on to Heidelberg, I think. And Deepa and Murdula undertook a 300-day mutation accumulation experiment in E. coli. In case you're not familiar with mutation accumulation experiments, basically a very dilute ancestor is streaked out over an agar plate. There's a pre-marked position, and whichever colony grows closest to that pre-marked position, that colony is sampled and streaked out over a plate the next day. And there were daily transfers in this MA experiment for almost a year over many, many lines, I think 30 to 80 lines in replicate. So this was a huge amount of effort. And then, of course, samples are put in the freezer at various points, and then whole genome sequencing was done on the samples in order to isolate 80 strains of this wild type, we'll call it the wild type, that each contained just a single mutation. And in fact, in the data that I'll show you here, they were just single nucleotide substitutions. So this is like the measure of the distribution of fitness effects as you'd like it to be done. So the fitness was then measured in 12 different environments for each of these 80 single mutation strains to create a distribution of fitness effects of single mutations. To my surprise, and in fact to Deepa's surprise when she started this, there is actually not a comparable study. So no one else has done whole genome sequencing, single mutations, and built a DFE in this way. And then, as if that weren't enough, Deepa and Murdula repeated all of that again in a mutator strain. So the mutator that they studied was missing this DNA repair enzyme MUT-Y. And this changes both the transition transversion bias and the ATGC bias pretty dramatically. So here's the wild type in turquoise and the mutant in coral. Those colors will be used throughout the talk. And you can see that MUT-Y, or the delta MUT-Y mutant, is strongly biased towards transversions and GC to AT mutations. So the beauty of the MA protocol, of course, mutation accumulation is used to limit the effects of selection or reduce it. Of course, we can't entirely get rid of selection, but we want to make it as small as possible. So the aim here is to have a bottleneck of size 1 and really accumulate spontaneously occurring mutations. But this experimental protocol that Deep and Rudula undertook also disentangles the mutation rate from the mutation spectrum in this mutator, because we're really just taking the same number of spontaneously occurring mutations. We actually have, in the end, we had 80 wild type mutations and 79 mutations from delta mut Y. So here are some preliminary results. Well, the first results that we looked at pooled across all 12 environments. Here are the DFEs of the mutations. And you can see clearly that the mutator here in coral has a shifted DFE, and it's shifted towards more beneficial mutations. This was really a surprise. I apologize that this figure is kind of elongated. It makes it a little hard to see the difference, but I think you can convince yourself, even without the statistics, that these two distributions are going to be different. I'll also point out that in the wild type, we had some nearly lethal mutations. Of course, an MA experiment cannot look at lethal mutations. So we're looking at the DFE in the absence of lethal mutations, but we had some near lethal mutations. And all the results that I'm going to show you hold even if we just discard these mutations. So whether we include them or not, we see this beneficial shift. And let me show you what happens if we look at each environment in isolation. So here on the y-axis, we have the relative fitness. So these violin plots are violin plots of the DFE across the different environments. So we had LB, and then we had minimal medium with 11 different carbon sources. Again, coral is the mutant. So you can see right away that the DFE in the mutant tends to have a beneficial shift. The line is 1, so a selective effect of 0. The black dots are the means of these distributions. And in 9 out of the 12 environments, the coral star indicates that there was a significant increase in the mean of the distribution for the mutant. So again, beneficial shift of the DFE in 9 of 12 environments. And again, these long tails, even if we remove these long tails, that holds. Of course, you may argue that many of the beneficial or many of the selective effects that we saw were quite close to 0. And so given measurement error, are we sure about what's beneficial and what's deleterious? So we looked at the data in a different way. Rudula ran a bunch of technical replicates to estimate the noise in our fitness measurements. And we figured out that the noise was something less than 5%. And so conservatively, we recategorized all mutations as either deleterious, neutral, or beneficial, where neutral was any mutation that was within 5% of the wild type or ancestor fitness. And I think, again, you don't need the statistics to look at these bar charts and convince yourself that the beneficial fraction in the mutator is higher on average than the beneficial fraction in the wild type. So these results were pretty compelling, no matter how Deep and Rudula analyzed them. So Deep is a very careful, critical thinker. And so she looked at these data and looked at a lot of alternative explanations. So as I mentioned, the beneficial shift holds even when the near lethal wild type mutations are discarded. We wondered whether the mut Y ancestor had a lower fitness to begin with and therefore could accrue more beneficial mutations, but its fitness was only lower than the wild type in 2 out of the 12 environments. And then Deep and Rudula also created paired strains. So to completely eliminate the possibility that these effects could have been because of some difference in the background once mut Y is knocked out, they engineered the same mutation in each background. So the beneficial mutations that first occurred in the mutant were put into the wild type background and vice versa. And without showing you all the data, essentially the paired strains showed no significant differences in fitnesses so that the mutations had the same fitness in either background. And then we also looked in the wild type strain in particular, mutations that were transversions or GC to A2 mutations. If you just look at those classes of mutations, overall, they had a higher benefit. So they were more beneficial in the wild type, but they just didn't happen in the wild type as often as the same beneficial mutations happened in the mutant in the MA experiments. So this is more evidence that what's going on here is it's really the shift in the spectrum that's changing the DFE. And just to check some other boxes, we also looked and there were no differences between the two sets of mutations that we obtained in mutant and wild type in coding or non-coding regions of the genome, other loci in particular, or specific genes, or in the resulting amino acid change in the 80 mutations that we looked at. So then, Dief and Rudela were wondering whether this effect was specific somehow, that transversions and GC to AT mutations just tended to be more beneficial in their system, although it was across many different environments. And I wondered whether this effect could in fact be much more general. So to test that, we created an NK fitness landscape. So this is a very standard fitness landscape model. Many of you are probably really familiar with it. We have sequences of length 8, 20, or 200 nucleotides. In a typical NK fitness landscape, the nucleotides are 0s and 1s, but we, of course, made ACGTs. We had nucleotides so that we could have transitions and transversions or GC to AT mutations, etc. And we ran adaptive walks across these landscapes. So here I'll say these adaptive walks, this is strong selection, weak mutation regime. So we have a ancestor sequence that is initially random. We create a random mutation. The probability that that mutation is a transversion, for instance, is set by whatever we have set the ancestor bias to be. And then if that mutation is beneficial, it fixes with a probability proportional to its beneficial effect. So the difference between the ancestor fitness and the mutant fitness. And if it fixes, we assume fixation has occurred, that mutant becomes the new ancestor and the walk continues. So that's one step in the walk. Of course, it would be nice to do this with clonal interference and simulate a whole population instead of just simulating, in a sense, the consensus sequence. But computationally, this is infinitely faster. So at the moment, we've just looked at adaptive walks. By varying K in these fitness landscapes, I have to thank Roland for introducing, or at least mentioning, epistasis in rugged landscapes. We can increase epistasis. So we can couple each nucleotide epistatically to a very small fraction or a very large fraction of other sites in the genome. And I'll point out how rugged some of these landscapes are. For example, just at n equals 20 and K equals 8, there are about 7,000 distinct fitness peaks in the landscape. For the 200 nucleotide, it sounds like a short sequence, but then there would be something like 10 to the 120 different fitness points in the landscape. So these are fairly rich, complex, rugged landscapes with lots of peaks. So what we do is let the ancestor walk along, adapt for a little bit, and then stop, freeze the walk, and let the ancestor make 1,000 random mutations, for example, and compute the fitness of those 1,000 random mutations, and thus create a DFE for that point in the walk for the ancestor. And then we can also freeze the walk at that point. The sequence is the same, but we imagine there's a mutant who has the same sequence, but somehow has a different mutational spectrum. So we change the probability that a transversion occurs, simulate 1,000 new mutations, and create a new DFE. So does that make sense? And then we can continue the walk a little bit further. So here are some of the results, what they look like. Here's fitness increasing over the course of a walk. And I'll draw your attention to initially the fraction of beneficial mutations in the DFE would be 50%, and it drops quite quickly. Likewise, the selective benefit on average drops over time, and the deleterious fraction concomitantly increases. And I'm going to, right about here is where I'm going to show you some results in a second. So there's a lot to unpack in this figure, so bear with me. So let's take an unbiased ancestor. So for transversions, there are, what, 8 transversions and 4 transitions. And so an unbiased ancestor has a TV bias of 2/3. We take that unbiased ancestor, we let it walk for a little while. We let it walk until the fraction of beneficial mutations in the ancestor DFE is, for example, 34%. That would be this dark line here. So that would be this point here on our time graph. Then we create the DFE of the ancestor, and then we create a DFE with all sorts of different mutator TV biases to compare. So we set the new TV bias to be 20% transversions, create a new DFE from that ancestor sequence at that point, and look at the percent change in the beneficial fraction of the DFE, and there's no change. Then we let the ancestor walk a little bit further. So these colors indicate more highly evolved ancestors further along in the walk. And you can see even for well-adapted ancestors, if we change the TV bias in the mutant, we don't see any change in the DFE. However, these results completely change if we look at a biased ancestor. So here's an ancestor wild type with a TV bias similar to what we have in the experiment. So, and then if we repeat this, you can see that even very early in the walk, when 34% of all mutations are still beneficial, we start to see that a mutant that reverses the TV bias has a larger beneficial fraction in the DFE. And this effect just increases dramatically as the walk continues. So there's a substantial beneficial shift in the DFE for mutants that can reverse the bias. And that was for for 2, I showed you 2 possible wild type or ancestor TV bias. So those would have been 2 lines across this plot. And we can repeat this for all possible wild type and all possible mutator bias values. And you can see here, there's a 0 along the diagonal, along the line of identity. There's no change. But in general, anytime that the mutator reverses the ancestor bias, there's an increase in the fraction, the beneficial fraction. And similarly, there's a change, an increase in the mean beneficial effect of beneficial mutations. So these results were really robust. They held across all the sequence lengths, all the degrees of epistasis that we tested. We saw qualitatively identical results for GCAT bias, which I won't show you here. And as I mentioned, the pattern holds for wild type sequences who are very far from an optimum. They have a lot of adaptive potential. 30% of their mutations or more are still beneficial, or for very well adapted wild type sequences. And so that was data to theory. And now I'm going to go back to some data. So this thought that this effect might be more general than we had expected, and that it might have to do with reversals in bias. This led us to do a little bit of phylogenetics, which I will run over very, very quickly because I understand so little of it. But I can give you the basic ideas. This was done by Gaurav Dian, who's in Heidelberg. So we looked at 11 bacterial DNA repair enzymes who change transversion or GCAT bias. We identified orthologs of those repair enzymes in 1,100 extant bacterial genomes, and then built a phylogeny and inferred where these enzymes had been gained or lost across the phylogeny, and therefore estimated how the TV or GCAT bias had changed over time in these lineages. So this is a really busy figure. I won't try to talk you through it completely, but just to give you an idea, for example, here's the phylogeny, obviously. Here's the presence and absence of all of these enzymes in the extant species. Here's the predicted bias in the extant species. There's a legend over here. If we look, for instance, at the mut-M enzyme, these red dots, you can see that it's predicted to have been gained here, but somewhere along these lineages, it's lost, and then it looks like it was regained here. So this inference, I'm afraid I can't really answer if you have questions about how this worked, but the results of this is we looked at, well, we don't know really what the bias is in the ancestral sequences in the phylogeny, but we can see if the changes are in one direction or another. And so what Wahl found was that the changes in bias tended to reinforce the current bias only rarely. And instead, there were lots and lots of examples where bias was reversed over evolutionary time. In some cases, as many as 7 different reversals were seen in some lineages. And again, I don't think you need the statistics to convince yourself, but the p-values, we did some kind of random reshuffling of these events across the phylogeny, which was a little bit technically tricky because you can't lose an enzyme that you haven't gained before. But with the reshuffling, the p-values were pretty astronomical. So there are definitely more reversals than reinforcements of bias over evolutionary history in these lineages. So overall, I think this is really exciting for me. I feel like it opens up a whole new set of questions to look at. There could be wide-ranging evolutionary implications of shifts in mutational spectra, in particular reversals. And there's a bioarchive. Our bioarchive paper is there if you're interested. And you can probably see over my left shoulder, since I'm working from home, I have actually a print of this beautiful Klimt painting. And I wanted to leave you with that because I imagine, if the wild type has been picking beneficial mutations from one part of this tree for a little while, even for not very long, the mutant does very well to look at a different part of the tree and try to pick beneficial mutations from there. So I think this is all about the low-hanging fruit has already gone for a particular bias. So that's it. I will take questions.
Okay, so thank you for this very nice talk.